Hello, everyone. My name is Qian. So I am a postdoc in Guocheng Yuan's lab at the Dana-Farber Cancer Institute in Boston. So today I'm going to be talking about how we use single-cell data to and also integrate this between single-cell RNA-seq and also spatial single-cell data that are coming out right now to dissect the different variations. So so we all know that the spatial context, although it's not that well studied right now, but it's extremely important for understanding tissue function. Let's say take a mouse brain, for example. There are multiple levels of spatial information. At the tissue level, this is the mouse brain organized into different anatomical regions. And then if we take a slice of it in the middle, we can reveal further structures within it from which we can get different regions, such as the hippocampus here, the visual cortex in the outside. And also these have very specific spatial patterns, such as anterior, posterior. So those are on the kind of a macroscopic picture of the organizations of the tissue. And down in the very microscopic level, we also have determined the cell types that are, for example, contained inside each structure. However, the arrangements of these cell types are not that well studied, and these are kind of microenvironment structures, which absolutely require single-cell resolution spatial data to answer. So together, we need to have single-cell profiling technology to dissect the heterogeneity in both the cell types and also in the spatial space. So currently, there are two types of single-cell profiling technologies. And each has its own advantage and disadvantage. By far, the most popular technology is the single-cell RNA sequencing, which has been pretty successful in determining cell types, due primarily to the fact that they can measure whole transcriptome information at a single-cell level. So we can leverage thousands of genes for identifying cell types. The key limitation of single-cell RNA-seq is that it loses the spatial information because these techniques require the cells to be dissociated. And also there are other minor minuses such as dropouts that often get encountered in measuring particularly low abundance genes. So in order to preserve the spatial information while capturing gene expression, now there are works done using imaging-based techniques to quantify both single-cell resolution, spatial, and gene expression. And the way this works is the following. So these techniques are developed several years ago called seqFISH and MERFISH from different labs in Caltech and also from Zhuangxiao Wei's in Harvard. So essentially, these take kind of a combinatorial based approach to identifying transcripts, where each gene species is specifically labeled by combinations of different colors of fluorophore. So for example, in this case, each round uses several color fluorophore to identify limited number of mRNA species. But in the subsequent round, you wash the probes and design probes such that each probe is hybridized to another set of colors. And in such case, you can switch the colors around between hybridizations. And what is determining the mRNA uniquely is combinations of different colors. And this can be scaled up to identify, theoretically, over several rounds of iterations, let's say 6 rounds of 15,000 genes. But in practice, this has been demonstrated to work quite well for hundreds of genes. So they have a very good sensitivity of detection. But the main thing is that they preserve single-cell spatial resolution, because these are actually done using microscopy imaging that preserve the cell context. So it becomes apparent that we need to integrate both modalities of data to better understand the cell type contribution and spatial contributions. And ideally, we would like to place individual cells onto this kind of two-dimensional grid, where one axis describes the tissue variation, the cell type variations, and the other axis describes the spatial variations. And each of these two components are essential for describing annotating cells. And we have developed some computational techniques for doing this. And more specifically, we focus on seqFISH data since they measure both expression and spatial information on the same set of cells. So the keyword is the same set of cells. So this is a microscopy image of the seqFISH. And we need to determine annotated cells with different colors that identify the cell types. And this, we can use the cell type signature from single-cell RNA sequencing data. And we can also identify variation using spatial seqFISH data alone. So this is a sort of like a spatial clustering picture showing how using the spatial relationships between the cells as well as the gene expression, what are these domains of gene expression patterns out there. And naturally, you can think that doing each of the decomposition will require a different set of genes. The cell type will require cell type markers, obviously, and the spatial variations would need to use genes that are kind of spatially coherent. So I'm going to decompose it into two components. The first is how do we identify cell types using information from RNA-seq. So we pose this as a classification problem where we learn a classifier of cell type based on RNA sequencing data. So this is the 8 major cell types from an single-cell RNA-seq. We identify the cell type markers that are differentially expressed between cell types. And then we train an SVM model per cell type, which essentially learned the weights that is being placed on individual genes. And the summary of the score kind of can help us determine on a 1 versus res model the probability that this gets assigned to a particular cell type. And then we apply this on the normalized seqFISH data. So we can repeatedly build 1 versus res models for each individual cell types that we are trying to predict. And then the maximum probability class would be the final class gets assigned to each cell type. So that is a pretty conventional approach. So I'm going to spend like 1 or 2 slides talking about spatial pattern detection because this is one area that we spend a little more effort on. So this is so the problem can be posed as the following: given spatial gene expression, what are these domains of gene clusters by expression? And this is different from just typical k-means results because k-means does not capture the spatial relationship between the cells. And if you simply do k-means, the resulting spatial pattern could be very, very noisy. So that is no good. So towards this end, we developed a hidden Markov random field model where the state of each cell is a balance of 2 kind of factors. One is the cell's own expression, and then also the neighborhood or the neighboring cell's expression, which can also influence the state of the cell. And the assumption is that these cells are not randomly scattered, but rather the neighbors' expressions tend to be similar. So this is the more in details how the HMRF works. We first build a neighborhood graph where we specify the spatial relationship. Each edge connects 2 immediate neighbors within a certain radius that we are giving. And then we build models to capture both the extrinsic factor, which is the neighborhood influence, the intrinsic factor, which is the influence of its own expression, cell's own expression. So this is the probability of class Ci, given the node Si, which is in the middle, and also the class configuration of neighborhood. We use the POT model to kind of model the energy functions that is on place Si. So the POT energy model essentially kind of decomposes the energies into a pairwise energies, which looks at the kind of the compatibility between each cell and its neighbor. If they have the same class, then we award such interaction. And if they don't, we assign to a 0. And we can control the weighting of the neighborhood using this beta parameter, where beta to 0 essentially just removes all the neighborhood influence. So this is the extrinsic factor, which determines based solely on the neighborhood. The intrinsic factor is simply determining the the probability of generating expression xi given a certain multivariate Gaussian distribution. And finally, to calculate the joint probability of both factors, we simply multiply them and normalize the probability value. So we apply this approach to study seqFISH and single-cell RNA sequencing data on the same tissue, mouse visual cortex, with over 1,500 single cells and 125 genes being measured. So these 125 genes from seqFISH include genes that are cell type variable genes, 25 of them, and the rest form some spatial patterns across the whole mouse brain. And the single-cell RNA-seq, we simply use existing data that is already published, the Tazek et al., which contains 1,800 single cells, 8 major cell types, which are listed as follows: the glutamatergic neurons, GABAergic, astrocytes. The latter 4 are the glial cell types. So the first set of results is on the cell type prediction. We noted that the seqFISH and single-cell RNA-seq correspond very well in terms of the average, the cell type average expression for all the cell type marker genes. In fact, the correlation is more than 0.8 in terms of Pearson correlation. So here, the special consideration here is how many genes do we need for prediction? Because here we're trying to predict 8 cell types, sorry. And obviously, if you try to predict more cell types, naturally you want to have more genes. So with 8 cell types, we do cross-validation within single-cell RNA-seq. And it turns out that we can already do 90% accuracy with only 40 differentially expressed genes, which are all listed here. But if we're trying to predict all 49 minor cell types in the study, then we would actually, this number increases up to 1,600 genes. And finally, we place a prediction onto this spatial position, and we noted that many of the cell types 9 cell types are quite scattered in arrangements. The blue here indicates the astrocytes, which is less abundant. And the red and the yellow here indicates the 2 neuronal cell types. So there isn't that much pattern that are shown here. So, however, we still want to seek alternative means to evaluate cell type prediction. So we turn to other staining, the Niso staining and DAPI staining. Niso staining has a pretty good property because they can stain neurons and not the glial cell types such as astrocytes. So we can overlay the prediction here. The magenta here indicate the glial cell types. And then we also do the Niso stains on the same set of cells. Being circled here, they could be very, very hard to see, but just take my words. These circles actually indicate the exact magenta position, indicating the glial cell types, and they don't have anything at all indicated by Niso. So this indicates that our prediction works pretty well. And for another slice of tissues, we observe similar things. So this is a validation. Spatial domains is the next set of results. So here, we limit ourselves to genes that are spatially coherent, and also we subtract cell type marker genes, realizing that some cell type marker can also have a spatial pattern, but to get a clean decomposition, we only work with those spatially coherent that are non-cell type genes. And we do a 9-state HMRF, and here, in terms of the spatial clustering, we get much clearer patterning. There are these patterns kind of distributed in terms of columns, which have individual evidence from individual genes, and also they have domain signatures. And then we next ask the question of what do these domains represent biologically? So we overlay the layer information. So those people who are familiar with the neuron, the neuronal visual cortex, they would know that the visual cortex is divided into 6 layers. And these layers seem to correspond pretty well with our domains. But we do further study of individual snippets of that picture. And over here is the layer information, and the colors indicate the domain. So it seems like our domain sit right at the boundary between layers. But what is more interesting is that if you pay attention onto the shape of the cells, seems to be a distinct association between cell morphology and the domains. So we do a quantitative analysis by extracting morphological features such as width, orientation of the cells, size of the cells, and also a Ferret parameter, and others of over 15 different features. And we can actually see quite a few morphological features that uniquely identify each domain. So where, on the other hand, if we overlay the cell type annotation on the same, they fail to explain the variations between layers, indicating that there are further variations that need to be captured by studying the spatial patterns. So finally, I will show how do we use the information between cell types and spatial domain information to dissect a common cell type called glutamatergic neurons. So indicated here are our glutamatergic predictions, over 800 of them. And they seem to divide into three groups. The groups of genes that are cell type specific, domain specific, these are expressions. And then also finally, there seems to be additional domain specific genes within the glutamatergic cells. And we apply them to reanalyze a single cell RNA-seq without any spatial information. And we observe distinct clustering of these cells when we do a simple k-means cluster on these 9 domain metagenes, indicating that the spatial cluster that gets recapitulated from single cell RNA-seq, even though we don't know what is the true mapping, but they seem to be expression clustered. So in conclusion, I have shown you how we can use the two modalities of data in order to borrow information from RNA sequencing to predict cell types on seqFISH data, and then also leverage seqFISH alone to predict spatial domains that we can use to reanalyze the existing single-cell RNA-seq data. And we illustrate this using the glutamatergic neurons example. And a version of this work is now on bioRxiv, so this is the link. You can check it out. And I also like to acknowledge my mentors, Guochen Yuan and Long Cai, who is generating the data, and also various funding sources. Lastly, thank the organizer of ISMB for allowing me to give a talk here. Thank you.
We have time for one question. If not, you could.
So
Yeah.
I was actually wondering, so if you look at the fish data, the seqFISH data, so every cell has an expression profile and also a spatial coordinate. So I was wondering if there's a correlation between spatial distance and expression profile distance. So if you only look at the data.
Yes, yes. So that's in fact the first step we are trying to do is to analyze the spatial patterns of individual genes, right? If there is a spatial correlation, then that should be displayed on that spatial pattern of that gene, right? If there's a cluster of them in space, then so we do have a score to capture that and use that for our spatial clustering.
Awesome. Okay, because I was wondering what if you just have that as a feature and then just only look at the seqFISH data, but we can talk offline about it.
Yeah, yeah.
Thank